Okay, class, so here we are, Monday, March 7th, no lecture exam today. We're going to try to kind of finish up the nervous system on chapter 8, and then we will start chapter 9 on our special senses. Most likely we'll have your next lecture exam a week from today. We'll see how far we can get through your special senses today. All right, so where we ended up with chapter 8 was talking about the meninges. And the meninges are three connective tissue layers that surround your brain and your spinal cord. And these three layers, no problem, Matt, I'm just getting your message. No worries. The three meninge layers are the same throughout the brain and spinal cord, and the dura mater is the outermost superficial layer, and it's the toughest of all the meninge layers. The dura mater forms two layers around the brain and only one layer around the spinal cord. The second meninge layer is very thin and wispy, and we call that the arachnoid matter. And then there will be a space between the dura and between the arachnoid matter, and we call that the subdural space. It's normally only a potential space containing a very small amount of serous fluid, and cerebrospinal fluid is, and blood vessels are found all in the subarachnoid space, which is the space below the arachnoid layer. And I'll show you a picture of this. And then the third meningeal layer is called the pia mater, which is very tightly bound to the surface of the brain and the spinal cord itself. So here we have our meninge layers. And again, these are the layers that surround your brain and spinal cord. They're just connective tissue. They'll be right underneath the skull. So they're kind of between the skull and between the cerebrum, kind of these three protective tissue layers, so that your brain isn't touching, directly with your skull bones. So here we have the dura mater, directly in contact with the skull. It's the kind of the toughest layer, and it's made of two layers. Here's the subdural space, which is right underneath the dura mater. The arachnoid matter is a little bit more thin and wispy, and underneath the arachnoid matter, you have a subarachnoid space where you have all your blood vessels and some fluid. And then you have the pia mater, which is the very tight kind of connective tissue covering that will kind of follow all the contours and curves and ridges of the brain itself. You can see here the dura mater, subdural space, arachnoid matter, subarachnoid space, and pia mater, and how all the meninge layers are the same as they kind of cover and keep your spinal cord safe as well. And this is a look at a cross-section of your spinal cord. You see the spinous vertebra and the body of your vertebral bones. Here's the cross-section of the spinal cord itself with the gray and white matter, and then you have the meninges kind of layered there as well, the pia mater, arachnoid matter, and dura mater, with the spaces in between the two. So those are the meninge layers. The ventricles, kind of the other part of the brain, is we have these ventricles, which are fluid-filled cavities within the brain. Only in your central nervous system, each cerebral hemisphere contains a relatively large cavity called the lateral ventricle. And then the third ventricle is a smaller cavity kind of in between the two, in the center of the diencephalon. It's located between the two halves of your thalamus and connected by foramina, or holes, to the lateral ventricles. So these are fluid-filled cavities, ventricles, that are all connected in the brain as cerebrospinal fluid flows through them. The fourth ventricle then is located at the base of the cerebellum and connected to your third ventricle via a narrow canal called your cerebral aqueduct. And again, I'll show you a picture of this in the next slide. The fourth ventricle will be continuous with the central canal of the spinal cord, so that cerebrospinal fluid then flows down your spinal cord. And your fourth ventricle also opens into the subarachnoid space, which is the space below your arachnoid meninge layer, so that the cerebrospinal fluid also kind of coats your brain and travels around the entire portion of your cerebrum as well. So here we have the brain ventricles, the two lateral ventricles, the third ventricle in between, and then the fourth ventricle you can see in yellow here. And you can see how they're all connected. This is how cerebrospinal fluid is created in these ventricles and then flows through them. The fourth ventricle is also opens up to the subarachnoid space. So you kind of have the cerebral spinal fluid, a continuous kind of cushion of water that also surrounds the outer portions of the brain. So your cerebral spinal fluid not only provides an extra layer of protection around the cerebrum, but when it fills up the ventricles within the brain, it gives the brain kind of a buoyancy-like factor. So your brain feels like it's floating. It's not so densely kind of just sitting there. The central canal then flows down the spinal cord with cerebral spinal fluid. And that's where you hear the word spinal taps, where they take out some of the cerebral spinal fluid to test it to measure some of your blood cell levels. So a little bit about cerebral spinal fluid. I already kind of got ahead of myself talking about it. It will bathe your brain and spinal cord. It provides a protective cushion around the brain and spinal cord. It's made from ependymal cells located in the choroid plexuses. And the choroid plexuses are kind of at the roofs or the top of all the ventricles. And that's what produce your cerebral spinal fluid. It will fill the brain ventricles, the central canal of the spinal cord, and also the subarachnoid space. So cerebral spinal fluid flows from your lateral ventricles into the third ventricle, down into the fourth ventricle, and then a small amount of it does go down your spinal cord, but the rest of it exits out through small openings in its walls to enter the subarachnoid space where it will surround your brain. Masses of arachnoid tissue called arachnoid granulations will penetrate the superior sagittal sinus, which is kind of like a dural venous sinus, so kind of a vein, a big vein, and cerebral spinal fluid eventually passes from the subarachnoid space into the blood through those granulations. So cerebral spinal fluid kind of bathes the vein, it cleans it, it gets away any waste material, and it eventually will go back into this venous sinus, so it'll get back into cardiovascular circulation as it kind of cleans out all the waste materials in the brain. So here is a good kind of picture showing the flow of cerebral spinal fluid. If we start at number 1, you can see how it's created in these lateral ventricles, and then it flows from 2 to the third ventricle, into 3, into 4, where some goes down into the central canal, and the rest of it travels in this subarachnoid space, which is labeled, well, the blue that you see labeled is actually the dural venous sinus, where the cerebral spinal fluid will eventually go into, but right below that blue layer is the subarachnoid space, which will be the layer of this kind of fluid cushion that just adds another kind of level of protection to the brain itself. Okay, and then cranial nerves, and we might end the chapter here. So your cranial nerves are 12 pairs of nerves that come off the brain and brain stem, and we name them with Roman numerals. And they kind of have 2 categories of functions. They can be sensor or they can be motor, and some of them are both, and we'll go through each of them now. The cranial nerve number 1 is your olfactory nerve to help with the sense of smell. So it's a pure sensory nerve. Cranial nerve number 2 is the optic nerve, also a pure sensory nerve for vision. Cranial nerve number 3 is the oculomotor nerve, which is a pure motor nerve for eye movement. So if you see it labeled as sensory nerve, it takes in information to the brain. And if it's labeled a motor nerve, it will send out some sort of response. So in this case, to control your eye movement. The trochlear nerve is also a pure motor nerve for one of your extrinsic eye muscles as well. Cranial nerve number 5, this is your trigeminal nerve. It's a really large nerve because it will actually have 3 branches. That's why we call it the trigeminal nerve. It will have both a motor and a sensory kind of function to it. So it'll be sensory for all pain, touch, and temperature of the eye and lower and upper jaws, and it will have motor innervation going out to control your muscles of chewing, like your masseter temporalis and your pterygoid muscles. Cranial nerve number 6, the abducens, is a pure motor nerve. It will control one of your eye muscles. The facial nerve is another sensory and motor kind of nerve mix. It's sensory for taste, so it helps you sense taste sensations on your tongue, but it controls, and that's why we call it the motor nerve, it controls a lot of your muscles of facial expression. So your zygomaticus, risorius, depressor anguli oris, and the orbicularis muscles as well. The vestibulocochlear nerve is a pure sensory nerve going to the cochlea in the vestibule, which we'll talk about in chapter 9, for hearing and equilibrium. Glossopharyngeal, whenever you see the word glosso, that has to do with controlling your tongue, and pharyngeal means it's going to your pharynx. This is also a mixed motor and sensory nerve. It's sensory for some of the taste buds on your tongue, and a motor to control the muscles that help you swallow in the pharynx. Cranial nerve number 10, this is a very special nerve, so I'm just going to put a star next to it, called the vagus nerve. It's both a motor and a sensory nerve. It's sensory and motor for organs in your thoracic and abdominal cavities. So all of your cranial nerves pretty much don't leave the neck and head region, except cranial nerve number 10 is the only cranial nerve that will innervate organs of your thoracic and abdominal cavities. Number 11 is your accessory nerve. It is a pure motor nerve for some of the muscles like your trapezius, your sternocleidomastoid, and muscles of the larynx. And then the hypoglossal is a pure motor nerve. Again, we see this glossal root stem of the word, so it will help to motor innervate your tongue. So these are the 12 cranial nerves, and in lab, I think you'll spend some time learning and labeling which is which. The olfactory nerve is easiest because it goes to the front. It's the most anterior cranial nerve because it'll go where your nasal cavity is to help you smell. The optic nerves crisscross each other as they go to the back of your eyeballs, and the place where they cross is called the optic chiasm. And then you see kind of all the other cranial nerves all bunched together coming out of the pons or the medulla oblongata. Another one that's really easy to note is your trigeminal nerve, because it's this large cranial nerve right here where you see the 3 branches coming off of it. The abducens is right below it, and then we have facial, vestibulocochlear, glossopharyngeal, vagus, and then the accessory nerve always looks a little unique because it comes off of the medulla oblongata, but it has several roots that are connected to it. So you'll spend some time in lab learning about those. The autonomic nervous system then, so we talked about your somatic nervous system innervates muscles that you can control, your skeletal muscles. Your autonomic nervous system innervates smooth muscle, cardiac muscle, and glands. And these are all things in the body that will work autonomically or automatically on a subconscious level. So without you know it, you have many autonomic functions of your autonomic nervous system that are maintaining homeostasis of your body subconsciously, so without you knowing it. And we divide your autonomic nervous system into the sympathetic division and the parasympathetic division, and we'll go through those divisions here. Increased activity in your sympathetic neurons generally prepares you for physical activity, so some sort of stressful situation or getting your body ready to move. That's your sympathetic nervous system. The parasympathetic stimulation generally activates involuntary functions like digestion that are normally associated with your body at rest. In the autonomic nervous system, there's always 2 neurons that extend from your brain and spinal cord to an effector organ. The first neuron we call the preganglionic neuron, and the second neuron we call the postganglionic neuron. And they're so named because your preganglionic neurons will synapse or connect with a postganglionic neuron in an area of autonomic ganglia, which are clusters of cell bodies of those neurons within your peripheral nervous system. The sympathetic division, so we'll talk a little bit more about that. Its cell bodies of the preganglionic neurons are in the lateral horn of your spinal cord gray matter, and they will come off between T1 and L2 segments. So we sometimes call your sympathetic division the thoracolumbar division because its spinal nerves are coming off between T1 and L2. The axons of preganglionic neurons exit through the ventral roots and project to either the sympathetic chain ganglia or other collateral ganglia that are nearby. Some preganglionic cell bodies of the parasympathetic division are located within the lateral part of the central gray matter, and they give rise to spinal nerves S2 to S4. Other parasympathetic nerves arise from cranial nerves, so we often call the parasympathetic division the craniosacral division. So you can see here that other cell bodies are located within the brainstem nuclei of the oculomotor facial glossopharyngeal vagus, and I'll show you a picture of this too. Axons of preganglionic neurons in your parasympathetic division will extend through spinal nerves or cranial nerves to terminal ganglia that are located very close to the effector organs in your head or embedded in the walls of the organs in your thorax, abdomen, and pelvis. And most of the thoracic and abdominal organs are supplied by neurons in the vagus nerve that are coming from your brainstem. It's one of your cranial nerves. So here we have a look at somatic and autonomic nervous system functions. First, to just review what the two are. Remember, the somatic nervous system is motor innervation to skeletal muscles that you can control. So you know when you're lifting your cup of coffee up to take a drink. The autonomic nervous system function, in contrast, is motor function that travels via a preganglionic and postganglionic neuron, because they go through a ganglion, to an area or organ of the body that is controlled subconsciously, so without you knowing it, like smooth muscle. You don't tell your digestive tract to digest your food. That happens automatically. So this is a good review of looking at then the parasympathetic and the sympathetic division. So on the left in blue, this is your sympathetic division of the autonomic nervous system. And you can see how the spinal nerves come off between T1 and L2, and the ganglia where they synapse on is called the sympathetic trunk. And then this is the parasympathetic division in pink on the right. And the parasympathetic spinal nerves come off in the cranial region using some of the cranial nerves, as well as some of the spinal nerves come off in the sacral region. So we sometimes call your sympathetic division the thoracolumbar division for the spinal nerves coming off of that region. And the parasympathetic division is the craniosacral division. And you can see here how, many of the nerves coming from, whether it's the sympathetic or the parasympathetic division, will innervate and control some of the same muscles, because if the sympathetic division, for example, increases your heart rate to get your body ready for activity, the parasympathetic division will decrease the heart rate. All right, and that is the end of this chapter. So I'm going to go ahead and end this, and we're going to kind of jump right in to chapter 9, which will be special senses. I'm just going to pull up this PowerPoint really quickly and take a quick sip of my water. Okay, and as you know, I don't really like to lecture for more than an hour because on Zoom and online, it's a little bit hard to pay attention. I figure an hour is enough for you guys. So this PowerPoint has about 80 slides, and I don't think we'll get through them all. So let's plan for your lecture exam, which will be on these chapters up through 9. Let's plan for that a week from today, and wherever we stop today, we'll finish on Wednesday. So just as your planning purposes, if any of you joined us late, your lecture exam will be a week from today. That's March 14th. But if you are in lab with me today on Monday, you will have your second lab exam. So be ready for that. Okay, so now we kind of end our nervous system talk, finally talking about special senses. And senses are a little bit more interesting to go through. So I promise this might be a little more interesting for you guys. The idea or the definition of a sense is any sort of ability to perceive a stimuli. So right now, you guys are being bombarded with stimuli. You hear my voice, unless you've turned me off because you're bored. with the screen. So your eyes are perceiving the stimuli from the pictures in your screen. You're perceiving stimuli from other things you hear if you have kids or pets in the background. So you are constantly being bombarded with stimuli, and your senses, the special senses, for example, give you the ability to perceive the stimuli so that you can understand what I'm saying, and that the screen that you're looking at, for example, the images you see are right side up, because all the images that come into your eye are actually upside down. And we'll talk about that. Sensation is the conscious awareness of stimuli received by sensory neurons. So you are consciously aware of what you're hearing because you can understand it, of what you're seeing. And you have many sensory receptors throughout the body. They're all different. They are sensory nerve endings that will respond to a stimuli by developing an action potential. So again, an action potential is like the electrical charge that passes through your nerves to bring that signal to your brain. So the images that are coming into your eye, kind of the light images that come in, that light gets changed into an electrical signal, an action potential that goes to your brain. The vibrations, the sound waves that are going into your external acoustic meatus, your ear canal, those sound waves will pass through fluid to turn into waves that vibrate your eardrum, bend your hair cells, that turn into an action potential, which kind of fires off an electrical signal to get that to the brain. Here is a classification of all of your senses. You can have general senses. The ones that are located in your skin, your muscles, and joints are called somatic, where they will help you detect touch, pressure, proprioception, temperature, and pain. Proprioception is the ability of your body to understand where it is in relation to each other. So proprioception helps you when you're moving your arms or running. You know, if you move your arms across your body, they don't touch each other. So proprioception is the ability for your limbs, joints, muscles, fingers to know where the other part of your body is in relation to each other. That's proprioception. And then also part of your general senses located in your internal organs are all the visceral senses that help to detect pain and pressure of something that could be going on in the inside of your body. And then your special senses have receptors localized within specific organs, like smell, taste, sight, hearing, and balance. So gustation, olfaction is taste and smell, sight is your eyes, and then hearing and balance occur in your ears. So the general senses, these receptors are over a very large part of the body that help to sense touch, pressure, pain, temperature, and itching. Somatic general senses provide information about the body and the environment, so anything external that your body is facing. And your visceral senses provide information about all your internal organs, so anything that's happening on the inside of the body. And then your special senses help with smell, taste, sight, hearing, and balance. And we'll go over all of those. We can have different types of receptors. Different types of receptors are mechanoreceptors to help detect movement. So an example of a mechanoreceptor would be a touch, pressure, or vibration receptor. Chemical or chemoreceptors help to detect chemicals like odors. So in your nose, you have chemoreceptors. Chemoreceptors are also found in your taste buds to help you taste the different molecules that have been dissolved on your tongue. And then photoreceptors detect light, and those are found in your eyes. Thermoreceptors detect temperature changes. You'll often find those in your skin. And nociceptors detect pain, and you'll find those in your skin as well. These are different types of touch receptors, and you might recognize some of these words from when we went over them in your integumentary system, because we see a lot of them in the dermis and epidermis layers of your skin where they should be. Your Merkel's discs detect light touch and pressure. Hair follicle receptors detect light touch. Meissner's corpuscles are deep within the epidermis to help localize tactile sensations. So they help you kind of perceive the ability to feel 2 points on your skin. Ruffini corpuscles are deeper tactile receptors to detect a continuous pressure in your skin. And then the Pacinian corpuscles are the deepest receptors. Those are usually associated with a tendon, a joint, to help detect even a deeper pressure, vibration, and position. So a lot of these sensory receptors, most of them, are found in your skin. And for good reason, because your skin is your body's first line of defense to tell your brain what is going on around it. So you'll see temperature receptors, touch receptors, pain receptors, deep pressure, vibration, position receptors, all found in the integumentary system, the dermis and epidermis of your skin. And that's where you want your sensory receptors. Your body's first line of defense should know what's happening around it at all times. And hopefully that picture looks a little familiar with what we've seen when we went over your integumentary system. Then pain. Pain is an unpleasant, as we all know, perceptual and emotional experience, but it's very necessary. If your body's receptors are not responding to pain anymore, that puts your body in a very dangerous situation. Pain is the way your body tells itself that it needs to get removed from a situation. Pain can be very localized, as you know, which can be a sharp, prickling, cutting pain, where it will kind of shoot off rapid action potentials, or it can be more diffuse, which is a burning, aching pain, and it has slower action potentials. And that sounds like a would you rather question, if you'd rather have localized or diffuse pain. I'm not sure what I would try or go for. Local anesthesia, it will send out action potentials to help suppress the pain, where it will kind of target receptors in local areas where chemicals are injected near the sensory nerve. So local anesthesia, think of getting dental work done. The dentist will shoot up just the nerve in the mandibular foramen. So it kind of just numbs the localized part of the lower kind of row of teeth if you're getting a root canal in your bottom row of teeth, for example. So that's a local anesthesia. Also common, sometimes they do that with kids before a shot. They'll just put a little gel or something so the kid won't feel the shot. General anesthesia is loss of consciousness. So when you think of someone going under, usually these are chemicals that affect the reticular formation, so the person will fall asleep. That's general anesthesia. Referred pain is very important to understand. And I know we have a lot of EMTs in here, so I'm curious, how much of this you guys learn in your EMT, or even if some of you are, firefighters, how much of you guys learn in this training? But referred pain originates in a region that is not the source of the pain stimulus. And this is really important to understand, especially if you ever work, as an EMT and you come to a site of an accident, or you ever work in an emergency room. Referred pain can be felt when internal organs are damaged or inflamed. And what happens is the sensory neurons from a superficial area of the body, so above the area, more superficial, and neurons of the source of pain itself will converge onto the same ascending neurons of the spinal cord. And I'll show you a picture of what I mean here. So these are areas of referred pain. So for example, if you have an appendicitis, that one isn't too bad. Normally the pain is felt pretty near the area of where it's originating. But what's happening is that the superficial region on the skin, kind of near your belly button, that superficial region is being supplied by the same sensory neuron that's coming from the sensory neuron that's innervating the appendix as well. So because they converge on the same pathway onto the spinal cord, your brain will perceive pain from an appendicitis or an inflamed appendix, maybe being a little more centrally located towards your belly button. And the big one here, as I'm sure you guys have all heard about, is the heart. And this is what's really important to remember. So pain from your heart, such as a heart attack, will be referred all along down kind of the inner left side of the arm, so that superficial regions of the arm will follow the same sensory nerve pathway to the brain as sensory neurons from the heart. So they're converging on the same pathway to go to the brain. So the brain perceives as pain from your heart, such as a heart attack, as coming from your left arm. So that's why a telltale sign of a heart attack is pain kind of shooting down the entire left side of the arm. Just because that's maybe the very common problem that occurs, a heart attack, that's often very known, but you can see other areas of referred pain: liver, gallbladder, the right shoulder, lungs and diaphragm, kind of the left upper neck, your esophagus, liver and gallbladder, kidney, stomach, and then the urinary bladder, ureter. So interesting, urinary bladder can be even referred to kind of the inner thigh region if anything is wrong with that. So that's referred pain. And then we'll go through your special senses. And again, we'll get through as much as we can here. Olfaction is one of our special senses. So these will have receptors located in kind of a special organ of the body. Olfaction itself is the sense of smell, and it occurs in response to odorants, which are chemicals that get dissolved in your nose. The receptors for these odorants are located in the nasal cavity and the hard palate. And we can detect over 10,000 different smells, which seems like a crazy amount of smells. I don't even know if I could think of 10,000 different smells. I feel like your height of smell is, or your sense of smell is heightened in certain situations, like pregnancy, which I'm going through right now. But we can detect a lot of smells, and you can see here, here's the olfactory nerve coming up to the nasal cavity, and it has fibers of the olfactory nerve that kind of are embedded within the nasal cavity, and they're embedded within kind of this mucus that will dissolve the chemicals from these odors you smell. And those chemicals that are dissolved from the odors will then get transferred or changed into an action potential. Then that gets sent up to your brain. So here's the olfaction process, very simple. The nasal cavity, as we know, contains a thin layer or film of mucus where all the odors are dissolved. Olfactory neurons are located in the mucus. We have little dendrites of these olfactory neurons, which contain cilia that help to pick up the odor, depolarize it, and then carry the odor to the cranial nerve number 1. And if you remember, and if not, that's okay, the frontal and temporal lobes will process the odor we smell. So here we have a neat picture showing how the dendrites of these neurons kind of end in cilia, which just provides surface area. They're embedded in the mucus layer of your nasal cavity to help pick up those chemicals. And then those chemicals eventually get picked up by these cilia and get changed into an action potential, which travels up through the olfactory nerves and then through the olfactory tract to the brain. Taste, also called gustation, is the sense of taste. Taste buds are the sensory structures that detect taste, and they are located on papillae on your tongue. They're also located on the hard palate, which is the roof of your mouth, as well as in your throat. Your taste buds, and I always like to describe this to students, the taste buds are not the bumps on your tongue. The bumps on your tongue are papilla, and then each papilla kind of has taste buds embedded within it. Inside each taste buds, there are 40 taste cells, and each taste cell will have taste hairs that extend into taste pores. So here we have the tongue. The papilla are the bumps on the tongue. And these bumps, as you can see, that's what you can feel. The bumps on the tongue are your papilla. But if we zoom in on kind of an area of a papilla, you can see the taste bud itself with all of the supporting cells, the taste hairs, and the taste pore. So again, the papilla are the bumps on your tongue, and kind of within the papilla are your taste buds. Taste process, your taste buds will pick up taste and send it to your taste cells. The taste cells will send taste to your taste hairs, and the taste hairs are what will actually contain the receptors that initiate or start an action potential, which is then carried to the parietal lobe to help sense the different tastes you're perceiving, and the brain will then process what you are tasting. These are the 5 different types of taste: sweet, sour, salty, bitter. Umami is from the breakdown of protein for amino acids, and some people describe umami as like a savory taste. I think of protein, breakdown of amino acids, think of like biting into a big juicy steak if you're a meat eater, and kind of that savory taste, or a hamburger. Certain taste buds are more sensitive to certain tastes, and taste is also very closely linked to smell. So if you have ever been to a winery, I know Southern California has lots of them. That's why to properly get the full sensation of the wine, you're supposed to smell it first before you taste it. So smell and taste are very intricately linked. Here's the pathway for the sense of taste. We see different cranial nerves actually innervating different parts of the tongue and different parts of the pharynx. So you almost have 4 cranial nerves innervating different parts of the tongue to help sense different taste sensations. And then vision. And vision and hearing are kind of where we'll spend the majority of the rest of the time, because these are kind of the huge sensory organs for vision and hearing, which are a little bit more complex. So first with vision, we have your accessory structures, which are your eyebrows and your eyelids and eyelashes. So your accessory structures are everything outside of the eyeball that still help with vision and keeping your eyes protected. Your eyebrows protect your eyes from sweat, and they also provide some shade from the sun. Your eyelids and eyelashes protect from foreign objects by rapidly blinking before anything can get in, as well as lubricating and kind of moving around the tears that are on the eye so that your eye remains clean with the moisture at all times. So here are the eye and accessory structures. You can see your eyebrows there, your upper and lower eyelids, the lateral and medial angle of the eye. And then within the eye itself, the white part you see from here is called the sclera. The colored part is the iris. So some of us have blue or brown, hazel, iris colors. And the iris is actually just a smooth muscle that controls the shape of the pupil. And the pupil is just a black hole. It's literally a hole in the center of the iris that allows light to pass through. And the iris will control if the pupil is constricted or dilated. Some more accessory structures. The conjunctiva is a very thin membrane that covers the inner surface of your eyelid. The lacrimal apparatus produces tears, and the extrinsic eye muscles help to move the eyeball. Here's the lacrimal apparatus, or lacrimal gland structures that produce the tears. You have little lacrimal glands kind of on each upper eyelid on the lateral side, and the flow of tears that come from the lacrimal gland kind of flows across the eye to the nose. So they go from lateral to medial as these tears kind of flow across the eye to keep your eyes clean. The tears will then empty into the lacrimal canaliculi, into the lacrimal sac, and then into your nasal lacrimal duct, into your nasal cavity. So your eyes are constantly keeping clean with tears. And this is why if you're crying a lot, you'll need a tissue, or you'll want to give someone a tissue, because the excess tears that are coming from the lacrimal gland are draining into your nose. Here are 6 extrinsic eye muscles, which control all the movements of the eye. And these 6 extrinsic eye muscles are all controlled via cranial nerve innervation. And they're all kind of labeled for what part of the eye they're attached to. So the superior rectus is attached to the top of the eye. The lateral rectus is attached to the lateral eyeball. The inferior rectus attaches to the bottom part of the eye. Medial rectus you can't see, but then the superior and inferior oblique are attached to the eyeball at an angle to all cause movements of your eyeball itself. So then we'll get into the anatomy of the eye. Your eye itself is a hollow fluid-filled sphere, and it's composed of 3 layers or tunics that we divide into chambers. So here we have the eye, and again, we'll spend time in lab kind of learning the anatomy of the eye, but let's start kind of with the 3 layers. We have the first layer is the most outermost layer is the fibrous tunic. That is made of the sclera, which is the white part of the eye, and the cornea is the transparent kind of concave structure that covers your pupil and iris. So that's your cornea. The vascular tunic is the second layer of the eye, and it's made of the iris, the ciliary body, which controls the shape of the lens, which is this structure, and the choroid layer, which is the layer where all blood vessels are. And then the third innermost layer, we call it the nervous tunic or the retina, and this is where you'll have all your photoreceptor cells that will help you perceive vision. If we kind of work our way to the front, then we can see this outer kind of cornea, which I'm just coloring with my red. Here we have the hole, that's your pupil with the iris around it. And here is your kind of large lens. It's a biconcave disc, which changes shape to help light to bend appropriately as it travels through then the vitreous chamber. And you see your optic nerve and how it attaches to the back of the eye there. So the fibrous tunic is the outermost tunic or layer of the eye. It consists of your sclera, which is the firm white outer part to help maintain your eye shape, provide attachment sites for muscles, and protect internal structures. The cornea is the transparent structure that covers your iris and pupil. It's kind of where you would put a contact lens, and it will allow light to enter and will help to focus or bend the light. The vascular tunic is the middle tunic, which contains all of your vasculature or your blood supply. The choroid layer is the black part, which will contain some melanin and delivers oxygen and nutrients to the retina through the blood vessels. The ciliary body is kind of a muscular body that holds your lens in place, and suspensory ligaments come out of the ciliary body to help kind of control the lens too. The lens itself is a flexible disc that can change shape. It can bulge out or get flat, and that will help to focus light on the retina, the innermost tunic layer. The iris is the colored part of our eyes. It will surround and regulate pupil size because it's actually a smooth muscle. The pupil itself is just a black hole. It regulates the amount of light entering, and your pupils will be constricted if you're in a bright, sunny room or you step outside first thing in the morning. Your pupils will constrict, and your pupils will dilate at night. So in areas where there's not a lot of light, your pupils will dilate to try to take in as much light as they can as they're trying to kind of be aware and help your body be aware of the surroundings. So here we have the lens in the ciliary body. So here are the ciliary muscles of the ciliary body connected to suspensory ligaments that control the shape of the lens. So here we have ciliary muscles, again, surrounding the entire shape of the lens itself. And then the suspensory ligaments connected from the ciliary muscles to the lens to control that shape. The iris then is kind of made up of two different smooth muscles that controls the shape of the pupil size. You have circular smooth muscles that help to surround the pupil to constrict pupil size, and then radial smooth muscles will help to dilate the pupil size. So that's your iris, kind of made of two kind of groupings of smooth muscles. The nervous tunic is the innermost layer. Then it's made up of the retina, which is kind of composed of two layers. It covers the posterior 5/6 of the eye. The pigmented retina is the outermost layer, and that pigmented retina just helps to keep light from reflecting back in the eye, while the sensory retina contains all your photoreceptors, the rods and cones that contain the interneurons. The rods are photoreceptors sensitive to light. So rods will be very helpful for you to function in dim light. So if you go into a dimly lit room that's kind of dark, your rods will kick it to gear to help your eyes adjust. And you notice after a while, when you're in a dimly lit room, you can usually see better after a couple of minutes as your rods are kind of picking up as much light as they can to help you see. The rods are much more common than the cones. The cones then are photoreceptors that help to provide for color vision. And you have three types of cones. You have blue, green, and red. And you can be colorblind for one of these three types of cones. So some people are only colorblind for blue colors, for green or red. And that just what that does is maybe maybe that's just one color they can't see very well. So here's what a rod looks like, a photoreceptor, and it has the outer segment kind of looks like a rod-like structure to it. And this should say cone, sorry, because the cone itself that has more of a disc-shaped segment to it. So that's kind of how they get their names. The rods have more of a rod-shaped outer segment, and the cones have more of like a disc or cone-shaped outer segment. We have other pigments and pigment proteins. Rhodopsin is the photosensitive pigment in your rod cells. Opsin is a colorless protein in rhodopsin, and then retinal is a yellow pigment in rhodopsin that requires vitamin A. So these are just different pigments and pigment proteins that are in your eyes. The effects of light on rhodopsin, and you won't be necessarily tested on this, but light will strike the rod cell, the retinal changes shape, opsin changes shape, they dissociate from each other, and that changes the rhodopsin shape to help stimulate a response in the rod cell, which results in vision. The retinal detaches from the opsin, and then ATP is required to reattach the retinal to opsin and return rhodopsin to the original shape. So basically, these pigments will change shape in the rods to help you see. And this kind of goes you through these 6 steps. I don't think you're tested on this, but it just shows you how light kind of dissociates these pigments from each other, and that changes the shape to help you be able to see with this pigment. The retina itself, so rods and cones will synapse or connect with bipolar cells of sensory retina. Horizontal cells of the retina modify the output of rods and cones, and then bipolar and horizontal cells will synapse with ganglion cells, and ganglion cells of the axons will converge to form the optic nerve. And here is just a look at the retina, and this just kind of shows you the direction of light and how light comes into the eye. What light does, so light from all the images that you see comes in and travels all the way to the back of the eye first in this kind of pigmented cell layer. And then it kind of travels through the photoreceptor layer of rods and cones as an action potential through horizontal cells, bipolar cells, and eventually the direction of those action potentials gets picked up by ganglions and nerve fibers that will travel through the optic nerve to the brain. So that kind of is a really simplified look at how light occurs. The direction of light hits the back of your eye, and then action potentials travel through all these layers of cells to then be picked up by the optic nerve to go to your brain. A little bit more about the retina. The macula is a small spot near the center of the retina. The fovea centralis is the center of the macula where light is focused when looking directly at an object. And the fovea centralis is where there are only cones, so it's the ability to discriminate very fine images. And just give me 1 second. We're dealing with kind of a family thing here. I just need to respond to one quick message. Okay, sorry about that. So in general, the macula is kind of a depression within the center of the retina, but within the macula, we have this fovea centralis layer. And the fovea centralis layer only has cones. So what your eye will kind of do naturally is it tries to focus all the light so it hits the back of the fovea centralis exactly, because that's where it can see the best. The optic disc then is the white spot just medial to the macula, and the optic disc are where all of your blood vessels enter the eye and spread over the retina. The axons will then exit as the optic nerve. And because we have blood vessels entering here, there are no photoreceptors, and we call this the blind spot of the eye. And I'll show you a picture. This is this is basically a picture of the retina. And the next time you go to your eye doctor, ask him if you can see the picture that he's taken, because what he's looking for, he's making sure the optic disc is showing the blood vessels coming out of it. So this optic disc is where your blind spot is. You have no photoreceptors here, because it's where the optic nerve attaches and where all of the retinal vessels enter the eye. And then just lateral to that on each eye, you have the macula, and the fovea centralis in it, which is just a little size, it's about the size of the head of a pen. The fovea centralis is where you'll have most of your cones. So your eyes will naturally try to move to focus light so it bends or is refracted right on this fovea centralis area. And this idea of a blind spot in your eyes can be tested if you just Google blind spot test, and you'll see charts that look like this, where you'll have to cover one of your eyes, focus on the red dot, for example, while covering your right eye, and then bring the chart closer and away from you. And when you're about maybe 5 or 6 inches away from this chart, you'll notice that the little T or the cross to the right will disappear from your peripheral vision. And that means that in that 5 inches, that cross has been kind of detected by the optic disc or the blind spot. Then the chambers of the eye, the anterior chamber is located between the cornea and the lens, and this is filled with aqueous humor, which is more watery. And this aqueous humor, which is watery, helps to maintain pressure of the eye, refracts the light, and provide nutrients to the inner surface of the eye. The posterior chamber is then located behind the anterior chamber, which contains also the aqueous humor. And aqueous humor, again, also is more of the watery type of fluid. So the anterior and posterior chamber are either, and I'll show you where they're located. They're located between the cornea and the lens, and I'll show you a picture of that here. The vitreous chamber is then located in the retina region, and it's filled with vitreous humor, which is a jelly-like substance. So whereas kind of in the anterior part of the eye, it was filled with more water, in the posterior part of the eye, it's filled with this vitreous chamber, which is filled with kind of like a jello-like substance. And this jello-like substance is really important for helping to maintain pressure, holding your lens and retina in place, as well as refracting the light, so bending it so it hits the back of the retina where it should. So that's the vitreous chamber. Then the functions of the eye, and we'll just get through a couple of these functions, and then I think we'll pause here. So the functions of your eye, it functions a lot like a camera, where the iris allows light into your eye, which is focused on the cornea, the lens, and humors onto the retina. So the iris kind of brings in the light, and then that light or those images is focused or refracted kind of back on the retina where it should be. And when your light strikes the retina, it produces these action potentials that are then relayed to the brain. And light refraction, which is the bending of light, and the image focusing are 2 important processes in how we establish vision. So light refraction is the bending of light, and it tries to bend the images or light onto a focal point in the back of the retina, which is a point where all of your light rays converge. And this will always occur anterior to your retina, right in front of it, so that the object is always inverted or upside down. So images that come into the eye are always inverted or upside down. Then focusing images on the retina, we talk about accommodation, which is how your lens changes shape to become less rounded so that the image can be focused on the retina. And this accommodation is just the word to describe how your eyes can focus on images closer than 20 feet. And accommodation, the lens kind of loses the ability to accommodate as we age. So that's why as we age, some of us will need reading glasses. So here we have focusing by the eye and how your lens focuses images and how the lens will change shape to be either see things distant or see things near. So for example, if you're looking at a tree, more than 20 feet away, your lens becomes flat. The ciliary muscles relax, the tension in the suspensory ligaments is high, so the lens becomes flat, and it will kind of create this focal point anterior to the retina and invert the image of that tree on the back of the eye. When you look at something close up, so less than 20 feet away, the ciliary muscles contract, the tension in suspensory ligaments is low, and it makes the lens become much thicker. So you can see kind of how the focal point changes a little bit more, but it again inverts the image as it gets focused on the back of the eye. The neural pathway for vision, and what I might do is I'll stop here at neural pathway because we still have quite a few slides to get through for vision before we talk about hearing and balance. So I think I'm going to go ahead and stop my recording here.